Hello my dear lovely students. Once again I welcome you on this amazing platform of Physics Wallah, where I, Ayushi Agarwal, your botany expert, is going to teach you the next important chapter of plant physiology, that is photosynthesis in higher plants. This chapter is very important. Mainly it is conceptual based, interesting chapter, and if I talk in terms of NEET, then surely every year we see 2 to 3 questions are asked from this chapter. So please pay attention to this chapter and hope you understand this chapter well. So without wasting time, let's directly start the chapter, that is photosynthesis in higher plants. Starting with the introduction part, first you should know what is photosynthesis. How will you define photosynthesis? So the term itself explains its definition. Photo means light, synthesis means to produce. So photosynthesis is a physico-chemical, it's a biological phenomena by which plants, they prepare their own food in the presence of light, carbon dioxide, water, mineral and chlorophyll pigment. So how will you define photosynthesis? Let's see. How will you define it? So photosynthesis is a physico-chemical process by which plants prepare their own food in the presence of light, chlorophyll, CO2, water and mineral. During this process, you are synthesizing a substance. You are synthesizing glucose with the help of some simple substances like CO2 and water. Therefore, photosynthesis overall, it is an anabolic pathway. Anabolic means the process by which you produce a complex substance with the help of simpler substance. So the general equation of photosynthesis will become, I'm going to write a complete balanced equation over here. So 6 moles of CO2 when combined with 12 molecules of water in the presence of light and other chlorophyll pigments, then they produce a complex substance which is glucose. Along with glucose, oxygen gas is released as a byproduct. Along with oxygen, water molecules are also released. So in photosynthesis, there is one major product which is your glucose and 2 not major product, or you can say byproducts, that is oxygen and water. You know, release of oxygen is one of the most important reason that why photosynthesis is a vital phenomena. Yes or no? Correct? Okay, so what is photosynthesis? I hope it is clear. It is the process by which simpler substances like CO2 and water, they are joined in the presence of light. Along with light, nutrients are also required, minerals are also required to produce the complex substance which is food. Along with this, oxygen and water are released as the byproduct. Hence, it is an anabolic pathway. As you can see, from the simple substances, we are synthesizing a complex substance. Correct? Now, this process is endothermic. Endothermic means the process in which you have to invest energy. So in order to produce glucose, energy is invested. The plant is going to utilize energy in order to produce glucose. So therefore it is anabolic as well as it is an endothermic process. I hope what is photosynthesis is clear. Now, why it is important? Why we keep on shouting? Why the government is shouting that plant trees, do not cut trees? And why government is fining the people, taking fines from those people who are cutting down the trees, etc. Why trees are so important? Because these trees are the only one which can perform, which can convert light energy into chemical form. They are the only organisms on this earth who can perform photosynthesis. And photosynthesis is a very vital phenomena for 2 major reasons. Number one, it purifies the air. You know, oxygen is released as one of the byproduct, and oxygen is very vital for respiration, for combustion, and many other processes. So plants are the only ones who can take in CO2 and give back O2. So they are very much responsible for purification of air. Otherwise, second important reason is their ultimate source of food. Plants are the only one who can convert light energy into chemical form. So for these two reasons, synthesis of food and purification of air, your plants become very important part of our system, and photosynthesis becomes a very vital phenomena. I hope these 2 points, last 2 points written on the screen, are also clear. Now, moving ahead. Now you can see that I have written the equation directly, that 6 moles of CO2 combines with 6 moles of water, giving glucose, oxygen, water, etc. And I have already told you that light is required, water is required, so things. Question comes, who told ma'am? Who told me that these are the things required and this is how reaction proceed? Surely there must be scientists who have done great discoveries in the field of photosynthesis in order to deduce this equation. There must be some scientist who told you importance of CO2, importance of light, importance of glucose production, etc. So now we are going to see the history of photosynthesis. Now we are going to see some important discoveries in the field of photosynthesis. Okay, history of photosynthesis. Number one experiment which we are going to cover is the experiment performed by Joseph Priestley, known as bell jar experiment. This Joseph Priestley, he performed a very simple experiment and told that during photosynthesis, oxygen is released as a byproduct. Joseph Priestley explained the importance of air in plant growth, or importance of air or importance of oxygen which is released from the plants during photosynthesis. To conduct his experiment, Joseph Priestley used a bell jar, inside which he placed a burning candle, a live rat, and a mint plant. So let's see the setup and the experiment of Joseph Priestley. Okay, so this is a bell jar. Now what he initially did, this Joseph Priestley, he initially took a bell jar and inside the bell jar kept a burning candle. Along with this kept a live mice. Right? So this is a mice and this is a burning candle. Not keeping the mint plant initially. Right? After some times what he observed, that both the burning candle as well as the live mice died. The candle got extinguished and the mice died. He got surprised that why within the bell jar, what actually happened inside the bell jar that resulted in the death of the mice and extinguishing of the candle. So in order to understand that, he now kept a mint plant inside it. So after some times you will see that candle got extinguished, right? And this live mice will die. Now what he did, he took another bell jar, and now in this bell jar, along with a burning candle, along with a live mice, he also kept a mint plant. Now he observed that once he kept the mint plant inside the bell jar, he found that neither the candle was getting extinguished nor the mice was dying. and he concluded that surely while keeping or after keeping the mint plant, there is proper flow of gases, there is proper circulation of gases taking place inside the bell jar, which is letting mint plant also survive, candle as well as mice also survive. See how. Now for candle, for burning of candle, for combustion of candle, oxygen is required. This is going to intake oxygen and in return will release CO2. Same mice. Mice also for respiration will intake oxygen and will release CO2. So what happened in A setup? After some time, the entire oxygen inside the bell jar was consumed and there was no oxygen left for burning candle and for live mice. So because of the lack of oxygen inside the bell jar, after some time, both candle got extinguished and the mice died. However, if I talk about the second setup, he kept a mint plant. Mint plant now circulated the gases properly by intaking the CO2 released by burning candle and the mice for photosynthesis. In return, released oxygen gas, which was then taken up by the candle and the mice, which led the survival of all the three organisms or all the three structures which are kept inside the bell jar. This led candle also burning, mice was alive, and mint plant was also happy. Correct? So this is how Joseph Priestley explained the importance of air for plants as well as for candle as well as for the rat. Hope you have understood this experiment by Joseph Priestley. Very simple and crisp experiment. Now proceeding further with the second experiment performed by Jan Ingenhousz. Jan Ingenhousz performed a thistle funnel experiment. What he did, or also known as inverted funnel experiment. Now in this experiment, Jan Ingenhousz depicted the importance of sunlight for photosynthesis. So Jan Ingenhousz explained the importance of sunlight with the help of his inverted funnel experiment. Let's see what is that experiment. So we are going to make two setups as was done by Jan Ingenhousz himself. So in setup A, what he did, he took a beaker which is full of water. Along with beaker, he kept a aquatic plant, maybe hydrilla, and he then covered this hydrilla with an inverted funnel. So this is the inverted funnel, which is actually covering your aquatic plant hydrilla inside a beaker which is full of water, right? So similarly, he made one more setup. So this is the A setup, for example. Now second is the B setup. Same, a beaker full of water, kept a hydrilla plant, covered itself with a inverted funnel. Now what is the difference? In A setup, he kept this under sunlight, whereas the B setup was completely kept in darkness. So A setup was kept in the light, whereas B setup was kept in the darkness. After some times, what he observed, Jan Ingenhousz observed that in A setup, air bubbles were rising, but there were no such air bubble in the B setup. These air bubbles are released, or these air bubbles are arising in A setup only because in the presence of light, this hydrilla plant is performing a process which is releasing gas, and when in water gas is released, bubbles start arising. However, in B, because there is no light, so such process is not able to be performed by the hydrilla plant, Now hydrilla plant is not performing photosynthesis, therefore there is no release of gas, and due to lack of gas there are no air bubbles. So in A setup, in the absence of light, no photosynthesis, no gas released, no bubble. Hence Jan-Ingen-House proved that sunlight is very necessary for photosynthesis. In the absence of light, there will be no photosynthesis. And this he proved by observing air bubbles. I hope both the experiments are clear, Joseph Priestley and Jan-Ingen-House. If yes, then we are moving ahead with the next experiment performed by Van Niel. Basically beta, Van Niel did a comparative study. He tried to compare the photosynthesis of green plants, that is higher plants, and the photosynthesis of purple and green sulfur bacteria. If you remember the chapter Monera, there I told you that some bacteria are photolithoautotrophs. That means they can perform photosynthesis by utilizing hydrogen sulfide as a reducing agent. As a result, they release sulfur gas instead of oxygen. Therefore, such bacteria, such photolithoautotrophic bacteria performing photosynthesis is known as anoxygenic photosynthesis. So basically Van Niel tried to compare the photosynthesis of these photolithoautotrophs, that is purple and green sulfur bacteria, and higher plants. When he was comparing the photosynthesis of both the organism, he observed one thing. He observed that by changing the nature of the reducing agent, the nature of the gas released also get changed. Let's see how. Chalo. So, what is written on the board? Van Niel, he compared the photosynthesis of purple and green sulfur bacteria with higher plants and found that oxygen gas is released from water but not carbon dioxide. I'll tell you about this equation. Chalo. So let's start. First, I'm going to write the equation of photosynthesis performed by higher plants. You know the equation is CO2 plus water. Water is going to act as a reducing agent. How? It is going to reduce CO2 into glucose and itself will get oxidized to oxygen, plus water will be released as a byproduct, which right now I am ignoring. So this is the photosynthesis of plants. Now let's try to write the photosynthesis equation performed by photolithoautotrophs, that is purple and green sulfur bacteria. Their equation is CO2. Now concept is the same. CO2 needs to get reduced, but the reducing agent in such bacteria is not water. Rather, it is hydrogen sulfide, inorganic compound, hydrogen sulfide. Hydrogen sulfide will reduce CO2 to produce glucose and itself will get oxidized to produce sulfur gas. So this is the photosynthetic equation of bacteria. Now what do you observe? You observe only one change. You observe only one difference between the photosynthesis of plants and bacteria. What is that? It is the nature of reducing agent. You see, the nature of reducing agent is different in both the types of organism, and as the nature of reducing agent is different, the gas released is also different. When water is the reducing agent, oxygen is coming out as a byproduct. When hydrogen sulfide is a reducing agent, then sulfur is coming out as a byproduct. So Van Niel concluded after observing the rate of photosynthesis, then surely reducing agents are the one who decides the gas produced. If it is water, oxygen. If it is hydrogen sulfide, then it is sulfur. So he wrote a general equation for photosynthesis, that is CO2 is common, carbon dioxide is the major substrate which will be reduced by any reducing agent. A means any inorganic element you can add. So CO2 will be reduced to glucose, and the nature of gas will be then decided by the inorganic element taken inside the reducing agent. Correct? Yes or no? Okay, so this is how Van Niel tried to show that water is the one responsible for oxygen, not CO2. However, later the concept of Van Niel was experimentally proved by two different scientists, Rubin and Kamin. These two people experimentally confirmed that yes, water is the source of oxygen for photosynthesis, not CO2. How? They actually doped the oxygen of both CO2 and water with the isotopes of oxygen O16 and O18. Chalo, let's see what they did. So what Rubin and Kamin did, they just doped the oxygen of CO2 and water both by using the isotopes of oxygen, which are O16 and O18. Suppose Rubin and Kamin doped CO2, oxygen of CO2 with O16. So now oxygen of carbon dioxide is being labeled with O16 and the oxygen of water is labeled with O18. Correct? So water oxygen is now O18 and CO2 oxygen is O16. If Van Niel is correct, if water is the source of oxygen, then the product side oxygen will be of which nature? O18. And if Van Niel is wrong, that is, if CO2 is the source of oxygen, on the product side, oxygen will be of O16 nature. This was the simple concept used by Rubin and Kamin. Now, complete photosynthesis was performed. Glucose is produced. And not surprisingly, Rubin and Kamin found that oxygen which is released is of O18 nature, thus proving that water is the ultimate source of oxygen, not CO2. Hope this experiment list is also clear to each and every one of you. Now, moving ahead with the next section. Experiment by T.W. Engelmann. This person is also very important. He actually proposed the first action spectrum. Now, what is action spectrum? If I try to explain in simple words, T.W. Engelmann with their experiment tried to show that which region of white light performs or helps in maximum photosynthesis. You know that the sunlight is comprising of so many regions, but the one which is useful to us is the visible spectrum, that is generally called as white light. Now, white light is having how many components? 7 components: Vibgyor, violet, indigo, blue, green, etc., etc. Now, T.W. Engelmann was very curious to know that whether all these 7 regions, that means all these 7 regions of visible spectrum, are they capable of performing same rate of photosynthesis? If yes, then it's okay. If no, then which region of white light is performing maximum photosynthesis and which region is not? So, with the help of T.W. Engelmann experiment, we came to know that which region is maximally utilized by the plants for photosynthesis. Now I am going to decode the experiment over here, and then I will explain you what is action spectrum. Clear? Shall we start? Chalo. Now there are some requirements for TW Engelmann experiment. First and basic requirement is a prism. So first I am going to draw a prism. You know what is a prism? Prism is a crystal through which if you pass the white light, then white light is going to split into its 7 components. So with the help of this prism, TW Engelmann passed a white light. Which now got split into its 7 components. That is VIBGYOR. 3, 4, 5, 6, 7. That is VIBGYOR. So these are the 7 components of the white light. Now through each component of white light, this TW Engelmann tried to grow a green algae. Why green algae? Because green algae is now going to perform photosynthesis by utilizing the light which you are going to provide along with other essential requirements. So in front of each and every component of light, Engelmann grew a green algae. So here, chalo, I'll draw it with the help of green color only. So he tried to grow green algae in front of every component of light, just to ensure that photosynthesis is being performed. Correct? Okay, so green algae is being grown in front of each component of white light. Now how Engelmann will realize that whether photosynthesis is being performed or not? By calculating the rate of oxygen evolution. Correct? Now rate of oxygen evolution we can only detect by growing some oxygen seeking, some aerobic bacteria in front of these green algae, because aerobic bacteria will flourish only in those regions which are richly supplied with oxygen. Otherwise aerobic bacteria will die. Correct? So TW Engelmann observed that aerobic bacteria very easily flourished richly in red region and blue region, and very poorly flourished in green region. So these white colored stands for aerobic bacteria. This shows that this growth of aerobic bacteria richly in red and blue region of white light shows that here maximum oxygen evolution is taking place, depicting that these 2 regions of white light are performing maximum photosynthesis. Correct? And in green region, very poorly bacteria are growing. That means in green region the rate of oxygen evolution is minimum, as it is not supporting photosynthesis. You know that green light is the one which is reflected back. It is not absorbed by the photosynthetic pigments, and that is why leaves majorly appear green in color. So with the help of this experiment by TW Engelmann, we came to know that there are 2 regions. एक second guys, just a second. VIBGYOR, right? Yeah. So with the help of this experiment, we can very surely make out that the 2 regions where aerobic bacteria are flourishing maximum, they are flourishing maximum due to maximum oxygen evolution. And this is happening why? Because red and blue region favors maximum photosynthesis. That means if you give whole white light but cut off red and blue, then there will be no photosynthesis. Or you can also say like this, you only give red and blue light to the plant, plant will happily perform photosynthesis even if you cut the other components of white light. And with the help of this experiment, he plotted a graph called action spectrum. Action spectrum means that telling the rate of photosynthesis is maximum in which region. So with this, he plotted a graph which is depicting the rate of photosynthesis is maximum in red and blue region. So for the graph, on x-axis, he plotted the wavelength of light, of white light, which is from 400 to 700 nanometers, 400, 500, 600 and 700. So this is the wavelength of light, and on y-axis he plotted the rate of oxygen evolution, right? And he simply found that there are two regions in which photosynthesis is occurring maximum, that is one in between 400 to 500 nanometers, that is blue and indigo kind of a region, violet and blue kind of a region, that is between 400 to 500 nanometers, and red region also shows high rate of photosynthesis, which is about at 700 nanometers. So the graph is going to have two peaks. This graph is going to have two peaks, one at blue region and another at red region. Otherwise the graph is going to be flat. These two peaks, one at blue region and another at red region, tells that here the oxygen evolution is maximum, telling us that the most favorable component of white light for photosynthesis is blue and red, and this is known as action spectrum. Clear? So T.W. Engelmann was the scientist who explained the first action spectrum, who explained that which component of white light are favorable for photosynthesis. Correct? Okay. So with this, I hope history is clear to everyone. All the experiments which I have told you so far. Now moving ahead, that where photosynthesis takes place. I am telling you that photosynthesis takes place, it is anabolic, endothermic, CO2 required, light required, so many things I am telling you, and all the time I am using the simple term plant. But where in plant does photosynthesis takes place? In root? Does it takes place in the stem or in the leaf? So now we are going to answer that question, that where does photosynthesis takes place. So simple answer to this question is that photosynthesis in plants takes place in their leaf. Leaf, they have special cells called mesophyll cells. Mesophyll cells represent the ground tissue system of the leaf, which contain a very special cell organelle called chloroplast. And inside the chloroplast, this process, this very important biological process, photosynthesis takes place. Clear? So first let's try to understand the structure of chloroplast, that now also where inside chloroplast also. Now you know inside the plant it is leaf, inside the leaf it is mesophyll cell, inside the mesophyll cell it is chloroplast. Now inside the chloroplast where photosynthesis takes place, to be more precise. And for that you need to understand the structure of chloroplast. Very easy, we have already done in the cell chapter, but let's try to revise it once again. So chloroplast, if I talk about, then chloroplast is a double membrane bound semi-autonomous cell organelle which is thought to be of prokaryotic in origin according to endosymbiotic theory. Yes or no? So this is my double membrane bound structure, right? There is one outer membrane and then there is inner membrane. The space between outer and inner membrane is known as intermembranous space. Correct? So the space between them is known as intermembranous space. The space enclosed within the inner membrane, this region, the ground substance is known as stroma. Stroma consists of DNA, which is double-stranded, circular in nature. They also have 70S ribosomes, plus they have so many other enzymes, majorly rubisco, to perform glucose formation. Apart from all these things, the other structures found inside the stroma are membrane-bound thylakoids. They have some coin-shaped membrane-bound structures called thylakoids. Usually, thylakoids are present in piles. They are placed one above the other. These piles of thylakoid is known as grana, which are interconnected to each other with the help of structures called stroma lamella or fret channels. Correct? Apart from this, they have double-stranded circular DNA, they have 70S ribosome in the stroma, enzyme rubisco, etc. So this is how the simple structure of chloroplast is going to look like. Now, where inside this structure will your photosynthesis actually take place? Right? Now beta, chloroplast shows division of labor. This cell organelle shows division of labor means it is going to divide its function into its different structures. Now, what happens? Overall photosynthesis takes place in two steps: light reaction and dark reaction. Correct? Light reaction, this part of photosynthesis is performed by the granule system, or you can say performed by thylakoids. So thylakoids or grana, they are specialized to perform the first half of photosynthesis called as light reaction. As on the surface of thylakoids, your photosynthetic pigments are located. So these photosynthetic pigments on the thylakoidal membrane easily trap the light and convert it into chemical form, that is your energy form ATP and NADPH2. Whereas the second half of the process, the photosynthesis, that is dark reaction, is performed inside the stroma, because the enzyme which is required for glucose formation is present in the stroma. So inside the stroma, CO2 is converted into glucose by utilizing the end products of light reaction. Hence you can say that chloroplast is showing division of labor. How? There is only one work with the chloroplast to perform photosynthesis. Photosynthesis is broadly done in two steps. First half is light reaction, second half is dark reaction. Light reaction is performed by thylakoidal system, whereas dark reaction is performed by the stromal system due to their specific reasons. So you can see the work is divided. The labor is divided in different structures of chloroplast. Clear? I hope the structure of chloroplast as well as the concept of division of labor in chloroplast is clear to each and every student. Yes, very good. Now, let's try to understand the concept of first light reaction, and then we will see that how the products of light reaction are used in glucose formation during dark reaction. But my dear students, before proceeding with the concept of light reaction, to perform light reaction, very important thing which is required are the photosynthetic pigments that are found on the thylakoidal membrane. Now you should know what are those photosynthetic pigments found in higher plants specifically. because there are so many ranges of photosynthetic pigments. But here we are studying photosynthesis in higher plants, so we should know that what are the list of photosynthetic pigments found in higher plants on the thylakoidal membrane of chloroplast. Correct? So to understand that what are the types of photosynthetic pigments, scientists perform different experiments, and the one successful experiment is paper chromatography that you can also perform in your schools. I hope you all must have performed. This is one of the most common experiment which students of class 12th or 11th are asked to do in their practicals, in their laboratories, etc. So paper chromatography, surely you must have performed in your school. So with the help of paper chromatography, scientists deduced or explained that there are 4 photosynthetic pigments found in higher plants. Correct? Now let's see. Chromatographic separation of the leaf pigments shows that there are 4 photosynthetic pigments, and the list of 4 photosynthetic pigments are chlorophyll A, chlorophyll B, xanthophyll and carotenoids. And all these 4 pigments, they have different colors. Chlorophyll A is bluish green in color. It is bluish green in color, whereas chlorophyll B is yellowish green in color. If I talk about xanthophyll, then they are purely yellow in color, and carotenoids are yellow orangish in color. So on the paper chromatogram, there will be 4 dots of these colors depicting each photosynthetic pigment. So how many photosynthetic pigments are found in higher plants? 4. Now, these 4 pigments, my dear students, can be categorized into 2 broad categories. That is, main photosynthetic pigments and accessory photosynthetic pigment. Main, accessory, what is this? Why it is? Why not all photosynthetic pigments are important? Why one is main and why 3 are accessory? So let's try to understand. Okay, so I told you there are 4 photosynthetic pigments which are broadly divided into 2 categories as main photosynthetic pigment and accessory photosynthetic pigment. Main photosynthetic pigment, also known as reaction center, is chlorophyll A only. Chlorophyll A is known as the main photosynthetic pigment or reaction center because it not only traps light of different wavelength but also have the capacity to convert that light energy into chemical form. However, if I talk about the remaining 3 photosynthetic pigments, that is chlorophyll A, xanthophyll and carotenoids, these 3 are accessory pigments. Accessory means additional pigment. How? They can trap light of different wavelength but cannot convert it into chemical form. They simply trap light and transfer it to chlorophyll A. Then chlorophyll A collects all the light and then convert it into chemical form. So main is chlorophyll A, accessory are the other pigments. Plant can survive only with the help of chlorophyll A but cannot survive in the absence of chlorophyll A. You may remove chlorophyll B, plant okay. You remove xanthophyll and carotenoids, plant okay. But if you remove chlorophyll A, plant is not okay. Because chlorophyll A is the main pigment. It not only absorbs light but also convert it into chemical form. But these accessory pigments, they only increases the efficiency of photosynthesis by trapping more light of different wavelength. but they simply trap light and give it to reaction center. Reaction center collect all the light and finally convert it into chemical form. So this is how the pigments are working in coordination for photosynthesis. Correct? Now beta, these pigments are not present independently. It's not like that on the thylakoidal membrane. Suppose this is thylakoidal membrane. Here is chlorophyll A, here is xanthophyll carotenoids. No, not like this. Rather, these group of photosynthetic pigments are present in packets. All the photosynthetic pigments, they are present in packets, and those packets of photosynthetic pigments are known as photons, or also known as photosystem. Photo means light, system means organization. So light trapping organization is photosystem, which consists of different photosynthetic pigments, and these photosystems are then present on the thylakoidal membrane. Clear? Yes or no? Okay, very good. So now you have understood that there are two types of photosynthetic pigments which are grouped under two broad categories as main and accessory. Now, before starting with the concept of photosystem, you have two different types of spectrum to study. One, I have already told you, that is action spectrum, that was given by T.W. Engelmann. T.W. Engelmann, you know, explained that maximum photosynthesis occurs in red and blue region of light. So, how he studied, you have already seen the experiment. He observed the rate of oxygen evolution, and with the help of that he plotted the graph which you can see over here. This graph is showing action spectrum. You can see on x-axis wavelength is being plotted, and on y-axis you can see rate of oxygen evolution, that is rate of photosynthesis, and there are two major peaks, one at blue region and another at red region. So this is action spectrum. Apart from action spectrum, there can one more spectrum called absorption spectrum. That tells you the different range that is preferred by different photosynthetic pigments. Like for example, if I specifically talk about chlorophyll A. So chlorophyll A is the main photosynthetic pigment that majorly absorbs in red region but can also absorb in blue region of light. So here you can see this is chlorophyll A. It absorbs in two regions, one in your blue region and another in your red region. Now talking about the next photosynthetic pigment, which is chlorophyll B. So chlorophyll B also absorbs in two regions, but majorly it absorbs more in blue region as compared to chlorophyll A, and it also have two peaks, one at blue region and another peak at red region. So this is for chlorophyll B. You can see one thing, that chlorophyll A peak as compared to chlorophyll B is higher at red region, and of chlorophyll B, if you check, then chlorophyll B peak is higher at blue region as compared to chlorophyll A. This is one of the best method to remember that which graph is depicting the photosynthetic range of which pigment. Correct? Apart from this, accessory pigments are your carotenoids and xanthophyll, which prefer only in blue region. So here you can see they have only one peak which is restricted only in blue region. So this is for your carotenoids, which are again accessory pigments. Now you can see that in absorption spectrum there are again only two peaks in two different regions, one in blue region and another in red region. Because photosynthetic pigments are maximally active at blue region and red region, that is why maximum photosynthesis also takes place only in red and blue region. That is why in action spectrum also you see two peaks, one at blue, another at red. In absorption spectrum also you see two peaks, one at blue, one at red. So we can conclude that both action and absorption spectrum are overlapping each other as they share common peaks, that is at blue region and at red region. So you can draw a third graph as well showing that action spectrum overlaps the absorption spectrum. Yes or no? I think this color is not visible. Yeah. So we can draw action spectrum overlapping absorption spectrum. Correct? Why? Because they both share the same peaks. Hope you have understood. Now we are going to actually start with the main process of photosynthesis, that is the first step called light reaction. Now before starting with the concept of light reaction, you should know the concept of photosystem, as I have already introduced this term before. Photosystem means group of photosynthetic pigments, right? Photo means light, system means organization. So different groups of photosynthetic pigments located on the thylakoidal membrane are known as photosystem. Each photosystem comprises of only one chlorophyll A, one reaction center and number of accessory pigments surrounding it. The main function of reaction center is to absorb light and convert it into chemical form, whereas accessory pigments are responsible for increasing the efficiency of photosynthesis by trapping light of different wavelength and transferring it to chlorophyll A so that light energy can be converted into chemical form. Correct? So how can we represent a photosystem? Let's check it out. Okay, so now here I'll show you the diagram of photosystem and then we will try to study the details of light reaction. So first let me show you photosystem. Right? So each photosystem comprises of one molecule of chlorophyll A, which is reaction center, that is surrounded by number of accessory pigments, also called as light harvesting complex, LHC. So these are group of accessory pigments whose function is to trap light of different wavelength and transfer it to chlorophyll A. This chlorophyll A collects the light and then convert it into chemical form with the help of different types of phosphorylation. Correct? So this is how photosystem looks like. Such photosystems are present on the thylakoidal membrane. There can be two types of photosystem depending upon the nature of reaction center. So how many types of photosystems are possible? Two types, that is PS1 and PS2. Correct? This types of photosystem is done on the basis of the nature of chlorophyll A, that is which wavelength of light is absorbed by chlorophyll A. On that basis there are two types of photosystem. In photosystem 1, the chlorophyll A absorbs light of beyond 680 nanometers. So it is also denoted as P700. Whereas in photosystem 2, the reaction center, that is chlorophyll A, can trap light of up to 680 nanometers. Hence, it is also denoted as P680 nanometers. Correct? So what is photosystem? Photosystem is represented by one reaction center and number of light harvesting complex which are made up of accessory photosynthetic pigments. There are two types of photosystems depending upon the nature of chlorophyll A, that is which type of chlorophyll A absorb which range of light, PS1 and PS2. PS1 is also called as P700 and PS2 is also called as P680. Correct? Now the naming, PS1 is called as PS1 and PS2 is called as PS2. This numbering is done on the basis of their discovery, not on the basis of their function. Functionally, PS2 is active first, followed by PS1. But since PS1 was discovered first, therefore it is numbered as 1. So you have to remember this thing. It will be asked in your NEET examination. NCERT very clearly mentions it, and yes, so you have to remember. Chalo, now let's try to read the points which are written over here, and then we will start with the main process of light reaction, where you will see that how plants with the help of photosynthetic pigments convert light energy into chemical form, that is ATP and NADPH. These end products of light reaction, you know, are further then utilized in the stroma of the chloroplast for synthesizing glucose in the second half of the process of photosynthesis, that is dark reaction. Right? Chalo, now let's read one by one. First, light reaction, also known as photochemical phase, because in this phase light is involved. So light reaction, other name for light reaction is photochemical phase, includes absorption of light, water splitting, oxygen release. All these steps you will know when I will teach you in details of light reaction. Correct? And production of chemical intermediates like ATP and NADPH. Second point, the pigments, the photosynthetic pigments are organized into discrete units called photosystems. So the pigments are organized into two discrete photochemical light harvesting complexes within the photosystem 1 and photosystem 2. So there are two photosystems 1 and 2. I've already told you. Next, these are named in the sequence of discovery. However, functionally, it is actually PS2 functioning first, then followed by PS1. The LHC, what is LHC? LHC are made up of hundreds of accessory pigments which are bound to proteins. So what is light harvesting complex? Represents the number of accessory photosynthetic pigments associated with protein. Right? Moving ahead. Each photosystems have all pigments. However, there is only one chlorophyll A forming a light harvesting system, also called antenna. Correct? Yes or no? Have you understood this? That each photosystem consists of one reaction center and number of accessory photosynthetic pigments which are known as light harvesting complex. Moving ahead. Now, these pigments, they help to make photosynthesis more efficient. I told you that accessory photosynthetic pigments, the main role is to increase the efficiency of photosynthesis by trapping light of different wavelength, and all the different wavelength of light which is trapped by light harvesting complex is then transferred to the single reaction center chlorophyll A for further chemical reactions. Next, the reaction center is different in both the photosystems, as I have already told you. PS1 is P700, whereas PS2 is P680 nanometers, depending upon the quality of wavelength of light which they trap. Correct? So now you know the concept of photosystem. Now we can actually start the process of light reaction. In plants, my dear students, there are two ways for operating light reaction. One is non-cyclic photo-phosphorylation and second is cyclic photo-phosphorylation. in non-cyclic photophosphorylation, you will see that how flow of electron is taking place in between the two photosystem on the thylakoidal membrane in order to generate ATP and NADPH2. Correct? So, first way of performing light reaction, which is most common in higher plants, is non-cyclic photophosphorylation. Non-cyclic, why? Because flow of electron between the two photosystem is done non-cyclically, and photophosphorylation it is known as because in the presence of light there is phosphorylation of ADP in order to produce ATP. Hence, this method of light reaction is known as non-cyclic photophosphorylation. You will see that the process of non-cyclic photophosphorylation gets a peculiar Z shape. When I will show you the flow of electron between the two photosystems, then you will observe that it takes a proper peculiar Z shape. Hence, non-cyclic photophosphorylation can also be known as Z scheme owing to its Z shape during the transfer of electron between photosystem 1 and photosystem 2. Correct? So now let's try to reciprocate whatever I have said right now on the board. So for this, first of all, I'll clear my screen. Right? Okay. Chalo. Now let's try to understand the mechanism of non-cyclic photophosphorylation. Where does it operate? It is going to operate on your thylakoidal membrane. So for that I am going to draw a thylakoidal membrane first. So where non-cyclic photophosphorylation operates? It operates on the thylakoidal membrane. So here is your thylakoidal membrane. Now I'll draw thylakoidal membrane in two layers so that it becomes easy for me to explain. Otherwise, thylakoid is a single membrane bound structure. Correct? But I'll draw it in two layers for my own convenience. So this is my thylakoid. Now, this thylakoidal membrane has photosystems present on itself. So this is thylakoidal membrane on which photosystems are present. Two photosystem 1 and 2. Photosystem 2 is present on the membrane but slightly towards the lumen, which is the space of the thylakoid. So this is the space of the thylakoid, which is known as lumen, and photosystem 2 is present on this membrane towards the lumen, whereas photosystem 1 is present on the thylakoidal membrane but facing the stroma. You know the space outside thylakoid is stroma, which is the ground substance of the chloroplast. So photosystem 1 is present on the membrane but facing the stroma. Correct? Now, this is how the arrangement of both the photosystems are there on the thylakoidal membrane to operate non-cyclic photophosphorylation, one of the major technique, one of the major way of light reaction, which is the first half of photosynthesis. Right? Now, what will happen? During daytime, sun will be there giving its 100% of light. However, its 100% of the light is not utilized for photosynthesis. Only visible range, and that too if I specifically mention, then red and blue range of light of visible spectrum of the sun is used for photosynthesis. Now this photosystem 2, which is also known as P680, this photosystem 2 is going to first accelerate the process of light reaction by trapping the light of its required wavelength, which is up to 680 nanometers. So now the first step is activation of photosystem 2 by absorbing light of its own requirement. Now PS1 has absorbed the light. Now, due to the gain of light energy, the valence electrons present in the photosystem 2, they will try to leave their shell because they have gained energy, they have become unstable, and now they cannot stay in their own system. So now electron from photosystem 2, the excited electron from photosystem 2 will leave photosystem 2 and it will be accepted by the primary electron acceptor present on the thylakoidal membrane called pheophytene. Now you see the transfer of electron has begun. Electron from PS2 has escaped and it is received by the primary electron acceptor of PS2, which is pheophytene. Now beta, electron is like a trouble. The electron carrier will accept it but will not keep it for long. Pheophytene might have accepted the electron from PS2 as it was required at that time, but now pheophytene cannot keep it for long. It will soon pass that electron to another electron acceptor which are present on thylakoidal membrane. So whatever electron is with pheophytene, it is going to pass that electron to another electron as well as proton carrier called plastoquinone, denoted as PQ. Beta, this PQ will accept the electron. Along with this, as I told you, it is a proton carrier as well. So it is going to accept the free proton from the stroma also. It is going to pick up the free protons from the stroma. After receiving the electron and proton from pheophytene and stroma, this PQ will get reduced. However, it does not want to remain in its reduced state. It wants to convert itself into its original form. So now what is it going to do? It is going to pass the electron and proton, whatever it has accepted, to some other acceptor. Okay, so now plastoquinone is going to pass its electron to another electron acceptor called cytochrome B6F. But cytochrome B6F, student, it is only the electron carrier. It refuses to accept the proton from plastoquinone. So what will plastoquinone do? Will it keep that proton itself or will it give back to stroma? No, both the things are not possible. So what plastoquinone does, unwillingly, unlikely, it is going to pass those proton in the luminal space of thylakoid. What plastoquinone did? It accepted the proton from the stroma, pumped in the lumen, accepted the electron from pheophytene, passed it to cytochrome B6F. Okay, now cytochrome B6 is having the electron of PS2. PS2 ka electron is there with cytochrome B6F. It has become excited. Will it try to remain in excited state? No, it is going to pass that accepted electron to another electron acceptor called plastocyanin on the thylakoidal membrane. Finally, the electron from PS2 has come to plastocyanin. Bachcho, meanwhile this transportation of electron is taking place, PS1 also traps light of its required wavelength, which is of up to 700 nanometers. And similar, on the same pattern like PS2, PS1 electron also becomes unstable and they try to leave their valence shell. So electron which is lost from PS1 is accepted by its primary electron acceptor called iron sulfur. Correct? Now, PS1 has given its electron to iron sulfur, because of which PS1 is having one electron less and iron sulfur is having one electron more. Okay. Now here plastocyanin is also having one electron extra. So what plastocyanin does, it passes that extra electron to PS1, satisfying the condition of PS1. Now you have observed one thing, that electron of PS2 has been transported to PS1 through various electron and proton acceptors located on the membrane. Meanwhile, electron from PS1 is with iron sulfur. It is also not going to keep it. It is going to pass to another electron acceptor called ferredoxine. Ferredoxine is also an electron acceptor, but it is not going to keep it for long. And finally, the electron from ferredoxine is accepted by the enzyme NADP reductase. What this enzyme is going to do? This enzyme is going to accept the electron as well as free proton from the stroma and will reduce NADP positive into NADPH2. Like this, you receive one of the end product of light reaction. So this is going to pass electron to the enzyme NADP reductase, which is going to reduce NADP positive into NADPH2. And now you have received one of your end product. Complete flow of electron has taken place. From PS2, electron has gone to PS1. From PS1, electron has gone to NADP positive. So this flow of electron is non-cyclic. Correct? Because you see PS2 is not receiving its own electron and PS1 is not receiving its own electron. So this is non-cyclic flow of electron in order to generate energy intermediates, chemical intermediates, ATP and NADPH2. NADPH2 you have obtained. Okay. Second thing is to see ATP synthesis to complete the process of non-cyclic photophosphorylation. Before we start with ATP synthesis, bachcho, one thing you can observe, PS2 is still lacking electron. Everybody is satisfied except PS2. PS2 is still saying, bhai, where is my electron which I have given to pheophytene? So, in order to overcome the deficiency of PS2, in the lumen of thylakoid, water splitting will take place. So now finally here, water splitting will take place to release protons, electrons and oxygen gas. Protons which are generated due to water splitting remains in the lumen. Oxygen, which you can see, is evolved as one of the byproduct of photosynthesis. You know, during light reaction, due to water splitting, there is release of oxygen gas. So here there is release of oxygen gas. Correct? And meanwhile, the electrons which are produced one by one, they are fed to photosystem 2. Correct? So now with this you can see what have you done to perform non-cyclic photophosphorylation. For non-cyclic photophosphorylation, there's non-cyclic flow of electron between the two photosystem, that is PS2 and PS1, with the help of various electron carriers in order to generate energy intermediates like NADPH2 and ATP. Meanwhile, this water splitting also takes place that releases oxygen, which is one of the byproduct of photosynthesis. This is how light reaction proceeds. Now you have to see concept of ATP synthesis. Concept of ATP synthesis was explained by Peter Michel. The hypothesis proposed by Peter Michel is known as chemiosmotic hypothesis. According to this hypothesis, in order to generate ATP, there are 4 basic requirements. Number 1 is a living membrane. So here you can see this thylakoidal membrane is the living membrane. First requirement is completed. Second requirement for ATP synthesis is proton pumps on this thylakoidal membrane. So you can see plastoquinone, it acting as a proton pump also. How? It is taking the proton from the stroma and pumping on the other side of the stroma, that is lumen. Now, due to the activity of such proton pumps, there is establishment of proton gradient. Proton gradient means that unequal distribution of proton across a living membrane. So due to the activity of plastoquinone, for example, the number of free proton in the stroma are going to reduce and their number is going to increase in the luminal space, right? Fourth requirement is the enzyme itself, that is ATP synthase. So, okay, so now finally we are going to see the location of the enzyme, that is ATP synthase. On the thylakoidal membrane itself, the enzyme ATP synthase is located. This enzyme is made up of 2 components. One component which you can see completely embedded, on the thylakoidal membrane, that is called as CF0 channel, and the second part you observe which is bulged out and it is bulging towards the stroma is known as F1 ATPase. These 2 components together they form ATP synthase enzyme. So this was the next requirement for ATP synthase, which is again located on the thylakoidal membrane itself. Correct? Now let's see what is the role of this enzyme and how ATP synthesis take place. So for ATP synthesis, as I already told you, gradient is must. There are 3 reasons during this non-cyclic flow of electron. There are 3 reasons to establish proton gradient. Number 1 is water splitting. What water splitting does? It pumps or it adds, it's not like pumping, the real term is adding. So when water splitting takes place, in order to satisfy the electron requirement of PS2, it releases 4 protons in the lumen, because of which the concentration of proton in the lumen as compared to stroma rises, creating a gradient. So number 1 reason is this, water splitting towards the luminal side. Second is the activity by plastoquinone. Plastoquinone basically picks up the free proton from the stroma and adds into the lumen, because of which again the number of free protons in the lumen are going to rise up. Third, the enzyme NADP reductase. This enzyme is going to accept the free protons from the stroma and produces NADPH2, because of which again the number of free proton from the stroma they get reduced. So there are 3 reasons how gradient is established. Now you can see overall proton concentration in the lumen rises up and in the stroma falls down. So what has been established? Gradient has been established. Now whatever excess protons are there in the lumen, they try to move out back to the stroma. There is only one way for the exit, that is through this enzyme. All the extra proton, with the help of F0 channel of ATP synthase, they try to cross the thylakoidal membrane. While they are crossing, they come in contact of next component of ATP synthase, which is F0 channel. Correct? Now, protons will come out, and once they come in contact with the second component of ATP synthase, this F1 ATPase is going to initiate phosphorylation. It will lead to the attachment of free phosphate to ADP in order to generate ATP while protons are moving out. So this method is known as non-cyclic. Flow of electron is non-cyclic. Photo, in the presence of light. Phosphorylation is taking place, addition of phosphate to ADP in order to generate ATP. This is the major way how light reaction is operating in the plant body to produce two very important chemical intermediates and oxygen gas. Correct? Yes or no? So this is how light reaction operates by the method of non-cyclic photophosphorylation, producing three major end products: oxygen that is released as a byproduct, ATP and NADPH2. They are produced in the stroma and they will be utilized in the stroma itself for producing glucose from CO2 during the next half photosynthesis, that is dark reaction. So this is the major way of Z scheme. Non-cyclic photophosphorylation is also known as Z scheme because of its peculiar shape that it obtains during the flow of electron between the two photo system. I hope whole non-cyclic photophosphorylation is clear. All the things like why it is known as non-cyclic, how ATP synthesis, how NADPH synthesis, what are the locations of photosystem, all questions are clear. Right? Okay. Second way is also there to perform light reaction. There you will see cyclic flow of electron in order to generate ATP. And that is known as cyclic photophosphorylation. This is not the major method of, this is not the major way of performing light reaction. Plants prefer non-cyclic method more, and there are certain reasons associated with it. Number one reason is that during cyclic photophosphorylation, there is no water splitting. Thus, there is no oxygen release. And you know, in photosynthesis, release of oxygen is also equivalently important. So cyclic photophosphorylation mein, there is no water splitting, there is no release of oxygen gas. Second, in cyclic photophosphorylation, only one chemical intermediate, only one energy intermediate, that is ATP, is synthesized. There is no synthesis of NADPH2. Hence, again, it becomes of less importance as both NADPH and ATP are required for dark reaction. So because of these two reasons, the preferred form of light reaction is non-cyclic photophosphorylation but not cyclic. However, still cyclic photophosphorylation operates, and there is a reason which I will explain you later on. But before that, let's try to understand what is cyclic photophosphorylation. In order to operate cyclic photophosphorylation, location required are either thylakoidal membrane or stroma lamella. You remember the two granules which are connected with the help of units called stroma lamella or Fred channels. So cyclic photophosphorylation can operate on both the structures inside the chloroplast, that is on the stroma lamella as well as on the thylakoidal membrane. First thing. Second thing, for cyclic photophosphorylation, there are only two requirements. Only photosystem 1 functions and ATP synthase enzyme functions. For cyclic photophosphorylation, there is no requirement of PS2 or there is no requirement of enzyme NADPH reductase. Clear? Chalo. Now, with this, let's start the cycle of photophosphorylation. Only photosystem 1 operates. So this is photosystem 1. There is sun during daytime. So photosystem 1 accepts light of up to 700 nanometers, because of which what happens? The electron, the valence electron of PS1, they get excited and they are lost, which are then accepted by the primary electron acceptor of PS1, which is iron sulfur. Right? Iron sulfur accepts the electron but is not ready to keep it. So it passes the electron to another electron acceptor, that is ferredoxine. Ferredoxine takes the electron from iron sulfur but is not happy to keep it. So now instead of passing to the enzyme NADP, ferredoxine will pass electron to plastoquinone. Plastoquinone will not only accept the electron from ferredoxine but will also accept the proton from the stroma, will pump it inside the lumen and pass the electron to cytochrome B6F, creating a proton gradient. Cytochrome B6F passes the electron as usual to plastocyanin, which again returns the electron to PS1, thus completing the cyclic flow of electron. Plastoquinone helps in establishing the proton gradient, which is then broken by ATP synthase in order to produce ATP. So during cyclic photophosphorylation, you see there is no requirement of water splitting as PS1 is already getting its electron. Second, there is no involvement of NADP reductase enzyme. So there is no synthesis of NADPH2. Only one of the chemical intermediate is being synthesized, which is ATP. So you will think that, ma'am, when cyclic photophosphorylation is neither releasing oxygen and in fact even not producing NADPH2, then why it is being operated? Let plant not operate this process, then why still it is being operated? And there is a reason attached to it, which I will tell you while teaching you the dark reaction, also known as light independent phase, also known as biochemical phase. Now I hope you have understood the concept of light reaction. You have seen that how on the thylakoidal membrane or on the stroma lamella, if I am talking about cyclic photophosphorylation, how light reaction operates, how water splitting takes place, how energy intermediates are produced. Now you are having energy intermediates in your hand. First half of the photosynthesis is done. Now let's talk about the second half of the chapter, second half of photosynthesis, that is dark reaction, also known as, also known as biochemical phase, also known as light independent phase. Simultaneously, beta, both light reaction and dark reactions are operating during the daytime itself. So don't think that if I'm talking about dark reaction, that means it is going to take place during night. No, even dark reaction is also going to take place only during day. Simply it is called as dark because it is not directly light driven. It is light independent. However, it utilizes the product of light reaction in order to fix CO2 into glucose. And dark reaction, this biochemical phase, takes place in the stroma of the chloroplast. Now we are going to see that how the end products of light reaction are used in the stroma of the chloroplast for glucose production. The mechanism of glucose production was explained by scientist Melvin Kelvin along with his supporters, along with his co-workers. This fellow Kelvin was actually working on green alga by using the radioactive isotopes of carbon, that is C14. With the help of radioactive isotope of carbon C14, this fellow Kelvin along with his co-workers explained the entire pathway that how CO2 is converted into glucose, and the name of the cycle is Kelvin cycle. Apart from this, This Kelvin cycle can also be called as C3 cycle. Why is it called as C3 cycle? I will tell you while I'll explain you the entire pathway. Now, this method of glucose formation is found in all photosynthetic plant, irrespective of they being C3, C4 or CAM. So, this is the universal method of how CO2 is converted into glucose. Right? Now, this fellow Kelvin divided the entire Kelvin cycle into 3 steps. Number 1 step is carboxylation, which is the most crucial step of the entire cycle. Second is reduction, and third step is regeneration. Regeneration is also a very crucial step in order to maintain the continuity of the cycle. Correct? Now, the enzyme which is required for CO2 conversion into glucose is rubisco. This is the most abundant enzyme present in the biosphere. It is very important, required for conversion of CO2 into glucose. The primary acceptor of CO2 in the stroma is a 5-carbon compound, that is RUBP, ribulose biphosphate. So 5-carbon RUBP combines with 1 carbon dioxide to produce the first stable product, a 3-carbon compound phosphoglyceric acid. Since the first stable product is a 3-carbon compound, that is why this cycle is also known as C3 cycle. Correct? Why? Because the first stable product in the C3 cycle is a 3-carbon compound, that is phosphoglyceric acid. Right? Now, my dear students, before I start with the entire cycle and try to explain you more about this cycle of glucose production, first let me give you some important information about this enzyme rubisco. Bachcho, this enzyme rubisco stands for ribulose biphosphate carboxylase oxygenase. That means this enzyme, suppose this is the enzyme rubisco, has a active site which is common for both the substrates, that is CO2 and O2. Enzyme rubisco shows competitive binding with CO2 and O2, and what substrate is going to bind rubisco is decided by the concentration of that substrate. Suppose if in the stroma the concentration of CO2 is more as compared to O2, CO2 will go and bind with the enzyme, and vice versa. However, if I talk about the affinity, the liking of the enzyme rubisco, then rubisco at low light intensity prefers binding with CO2, and at high light intensity it prefers to bind with oxygen. Correct? Mainly, this is how enzyme is. This is what enzyme is. It can bind with both CO2 and O2. But what plants require? Plants specifically require that CO2 should go bind with the enzyme to proceed with the further reaction. Right? Plants' requirement is for glucose production that CO2 should bind with enzyme, not O2. But you know, it is not in the hands of the plant. It is all in the hands of the concentration of both O2 and CO2. So if concentration of O2 wins over CO2, then oxygen will go bind with rubisco, will perform oxygenation to produce some different stable product that do not take participation in further C3 cycle, resulting in the fall of biomass production, and that is known as photorespiration. So what is photorespiration? We'll deal in more, but right now I have just given you a hint that this enzyme Rubisco has dual activity. It can also show oxygenation and it can also show carboxylation. If it is showing carboxylation, well and good, but if it shows oxygenation, then it leads to a wasteful phenomena called photorespiration. We'll talk about photorespiration, but first let's see what is C3 cycle and what are these three steps. So the number one step is carboxylation, that is binding of CO2 with the primary acceptor, which is RUBP, ribulose biphosphate, in the presence of the enzyme Rubisco to produce the first stable product, a 3-carbon compound phosphoglyceric acid, and of this 2 molecules are produced to balance the number of carbon on both reactant and product side. So basically it's a 3-carbon compound, 2 molecules of phosphoglyceric acid are produced. Now, since the first stable product is a 3-carbon compound, therefore this cycle is also known as C3 cycle. And C3 cycle operates in all photosynthetic plants for producing glucose. First step is done. Second step is reduction, where phosphoglyceraldehyde is reduced to 2 molecules of 3-carbon compound phosphoglyceraldehyde. In reduction step, the reducing agent is NADPH2, which you have obtained actually during light reaction. So see how the end products of light reaction are utilized in reduction step of C3 cycle. For 2 molecules of NADH2 are oxidized. In return, PGA gets reduced. This is also energy consuming step. So 2 ATP are also consumed. Again, ATP is also coming from C3, is also coming from light reaction. After this step, you obtain 2 molecules of phosphoglyceraldehyde, 3-carbon compound. So in total, there are total 6 carbon, correct? 6 carbon over here. Now these 6 carbon, if I want to bifurcate, then surely I can understand that 1 carbon is actually from CO2 and 5 carbons are from RUBP. Bachcho, for glucose production, we will only utilize the carbons of CO2. The carbons of RUBP you have to regenerate. You have to convert them back to RUBP. RUBP is only a helper. It is only helping, assisting CO2 to enter into process of C3 cycle. But you cannot utilize their carbon. In order to complete the cycle, regeneration of RUBP is very, very important to maintain the continuity of the cycle. I mean, if you're not going to regenerate this RUBP, then to what second molecule of CO2 will bind? So to maintain the continuity, regeneration is very, very important. And per RUBP regeneration, 1 ATP is required. Correct? So this is how the cycle goes on. Now, you know 5 carbon of PgAl has been regenerated to produce RUBP. Still 1 carbon is left and that carbon will be utilized for glucose production. But ma'am, glucose to 6 carbon compound hai and right now we only have 1 carbon in our hand. Therefore students, this cycle is going to run 6 times in order to produce 1 molecule of glucose. Right? Yes or no? So this is how C3 cycle operates. Now let's focus on its energetics. Per CO2, total number of ATP utilized is 2 and 1, 3 ATP. And along with 3 molecules of ATP, 2 molecules of NADPH2 are also consumed. But this energetics is with respect to per CO2. Now if question comes with respect to per glucose, so with respect to per glucose, the cycle is going to run 6 times, making 18 ATP utilization and 12 NADPH2 utilization. Yes or no? 3 6 are 18, 3 2s are 3 6 are 18 and 2 6 are 12. So 12 NADH2 molecule and 18 ATP are consumed for producing one molecule of glucose with the help of C3 cycle or Kelvin cycle. Right? Have you understood this? This is how dark reaction, this is how glucose production takes place in all photosynthetic plants. Now students, I already told you one mechanism, one extra wasteful phenomena by the name photorespiration. Photorespiration happens when photorespiration is basically a phenomena which happens when the concentration of oxygen in the stroma of the chloroplast is high. As a result, oxygen goes and bind with RUBP in the presence of the enzyme Rubisco in order to produce the first stable product, a 2-carbon compound phosphoglycolate. A 2-carbon compound, and along with this, one molecule of phosphoglyceric acid is also produced. So on the reactant side you have 5 carbon, on the product side also you have 5 carbon. But you know there is one trouble. For continuing with the Kelvin cycle, 2 molecules of PGA are required. But when oxygen binds, instead of 2 molecules, only 1 molecule of PGA is obtained and another compound by the name phosphoglycolate is obtained. This phosphoglycolate cannot enter into the cycle for glucose production. First, you need to convert this phosphoglycolate into phosphoglyceric acid, which is done with the help of 3 cell organelles, that is chloroplast, peroxisome and mitochondria. So with the help of these 3 cell organelles, this molecule, this 2-carbon compound is converted into 3 carbon. Then anyhow those 2 carbons, then those 2 molecules of phosphoglyceric acid will further proceed in C3 cycle. So can you see, this is some extra work has to be done, unnecessary, and in this, and while doing this conversion, ATP is also required. So the work which could have been done in a straightaway manner if CO2 was binding with Rubisco, but now due to binding of oxygen, time is being consumed, extra energy is being consumed, extra cycle has to be operated. Yes or no? So that is why photorespiration is simply a wasteful phenomena where overall biomass production, glucose formation decreases. But there is no escape for photorespiration in C3 plants. C3 plants cannot escape photorespiration. They have to operate this wasteful phenomena because it is all in the hands of the concentration of O2 and CO2, and which is highly variable. If oxygen wins with CO2, then same thing is going to happen. Phosphoglycolate will be produced, which again need to be converted into phosphoglyceric acid involving 3 different cell organelles and ATP. So photorespiration, you can see, is a wasteful phenomena. Okay, C3 plants cannot escape, but there are certain plants which grow in dry tropical region. The plants which are already facing high light intensity, water stress, soil salinity, etc. and etc. These plants, in order to make better biomass production, in order to avoid the trouble photorespiration, they produce different type of leaf anatomy. They produce different type of arrangement of cells in their leaf. So what I'm talking about, see, C3 plants cannot escape photorespiration. But what about the plants which are growing in dry tropical region? What about those plants who are already facing troubles like high light intensity, water stress, soil salinity? Will they also bear the pressure of photorespiration? No. So such plants are known as C4 plants. C4 plants are the special plants because they avoid photorespiration by showing some different arrangement of cells in their leaf. So now let's talk about the C4 plants and let's see how do they avoid photorespiration. So first thing I have told you that C4 plants, they grow in high light intensity, right? They grow under water stress, they grow under soil salinity condition, and they show some different arrangement of the leaf. That different arrangement of leaf cells in C4 plant is known as Cran's anatomy. Crans means wreath. That means arrangement of cells. So in case of C4 plants, if I talk about their leaf cells, then usually C3 plant have one type of cell, that is mesophyll. But in C4 plants, they have two types of cells, that is mesophyll cells and bundle sheath cells. Both the cells are concentrically arranged around the vascular bundle. Suppose this is my vascular bundle. Around this vascular bundle, several layered, thick-walled bundle sheets are present. Right? So they are thick-walled to avoid the exchange of gases. So these are bundle sheet cells whose stroma have Rubisco enzyme. So bundle sheet cells are several layered, thick, concentrically arranged around vascular bundle, impervious to gaseous exchange, bundle sheet cell, around which is there a layer of mesophyll cell whose cytoplasm contains a special enzyme called pepcase, phosphoenol pyruvate carboxylase. So these are mesophyll cells and bundle sheet cell. Now mesophyll cell, as I told you, what they have a special enzyme which is called as pepcase, but they lack Rubisco enzyme. If I talk about bundle sheet cells, then bundle sheet cells, they lack pepcase, but their stroma contains the chloroplast stroma contains the enzyme Rubisco. Okay, so this is how the leaf anatomy of C4 plants look like. So they have vascular bundles around which they have bundle sheet cell and around which they have mesophyll cell. Now both the cells will participate in glucose production. Both the cells will participate in basically photosynthesis, right? And they are going to perform photosynthesis in such a manner that they avoid photorespiration. Chalo. Now let's try to see that how do they do the entire mechanism of glucose formation. Suppose this is a mesophyll cell which is connected to bundle sheet cell with the help of plasmodesmata. So this is my plasmodesmata. These are mesophyll cells and these are bundle sheet cell. Now mesophyll cells, they are open to gaseous exchange unlike that of bundle sheet cell. So whatever gases come inside, they can easily enter inside the mesophyll cell. So mesophyll cell receives the gases. Now I told you that mesophyll cell, they have a special enzyme called pepcase. This peptidase enzyme has a power to carboxylate a 3-carbon compound phosphoenol pyruvate. So the primary acceptor in case of C4 plants, in case of C4 cycle, is a 3-carbon compound, that is phosphoenol pyruvate. So CO2 inside the mesophyll cell binds with its primary acceptor, a 3-carbon compound phosphoenol pyruvate, in the presence of the enzyme peptidase, to produce the first stable product, which is a 4-carbon compound called oxaloacetic acid. Since the first stable product is a 4-carbon compound, that is why this cycle is also known as C4 cycle. And plants, so since the first stable product is a 4-carbon compound, as I told you, that is why this cycle is also known as C4 cycle, and the plants exhibiting C4 cycle are known as C4 plant. This cycle was completely explained by 2 scientists, Hatch and Slack. So this pathway of glucose formation in C4 plants is also known as Hatch and Slack pathway. Coming back to oxaloacetic acid, which is a 4-carbon compound, it then converts into another 4-carbon compound called malic acid. This malic acid then travels to the bundle sheath cell through plasmodesmata. It is going to come inside the bundle sheath cell, right? Once it enters into bundle sheath cell, now this 4-carbon compound malic acid will break into a CO2 and C3 acid. C3 acid is going to go back to the mesophyll cell, and with the help of the enzyme PEP synthase, it is going to regenerate the primary acceptor C3 acid. With the help of the enzyme PEP synthase will help in the regeneration of PEP, and this step requires 2 ATP. So regeneration step requires how many ATP? 2 molecules of ATP are required. Now you can see in the bundle sheath cell you are having CO2, correct? Bundle sheath cells, first of all, they are thick walled, they are impervious to gaseous exchange. So whatever gases is coming inside the bundle sheath cell are coming through mesophyll cell. So normally gases are coming along with this due to the breakdown of malic acid resulting in the production of CO2. The overall CO2 concentration in the bundle sheath cell increases, and this increase in CO2 concentration always help to promote C3 cycle. Now in bundle sheath cell, the chloroplast, they have Rubisco enzyme. Now always CO2 concentration is going to be maintained high because C4 plant shows internal CO2 concentration mechanism. These bundle sheath cells, they themselves are manufacturing their CO2 by the breakdown of malic acid. So always inside the bundle sheath cell, overall the concentration of CO2 is higher as compared to O2. Hence, inside the bundle sheath cell, the CO2 is always going to bind with the RUBP in the presence of the enzyme Rubisco and will complete the C3 cycle to produce glucose. So now this will undergo C3 cycle to produce glucose. Now you see, even in C4 plants, for glucose production, C3 cycle is getting operated. Overall, it is C4 cycle, and C3 is a part of this C4 cycle. Correct? Yes or no? So as I already told you that be it C4 plant, C3 plant or any other groups of plant, C3 cycle is must to operate in them to show glucose production. Apart from that, they may have some additional mechanism like you see in C4 plants. They have some extra mechanism going on to increase the CO2 concentration inside the bundle sheath cell so that photorespiration can be avoided. So C4 plants, they do not show photorespiration just because inside their bundle sheath cell concentration of CO2 is always maintained high. Some examples of C4 plants are like maize, sorghum, etc. Sugarcane. Okay, now let's read this page. This page I have strictly taken from your NCERT. Let's try to read the lines. Plants that are adapted to dry tropical regions, they show additional pathway of glucose formation, that is C4. Correct? Now, otherwise, the main is Kelvin cycle for glucose production. C4 plants are special. Why? Because they have special type of leaf anatomy. They can tolerate higher temperature. They show response to high light intensity. Even during high light intensity, they do not show photorespiration. They lack a process called photorespiration, because of which they have a greater productivity of biomass. Moving ahead, particularly they have, as I told you, they show crans anatomy. Crans, which means wreath, right? In which they have two types of cells, bundle sheath cells and mesophyll cells, which are concentrically arranged around the vascular bundle. Bundle sheath cell forms several layers around the vascular bundle, and they are characterized by having large number of chloroplasts which are without grana, plus they have thick wall and no intercellular spaces. Correct? So please remember all these points. I hope the cycle of C4 plant and arrangement of cell in C4 plant is clear. It was studied by scientist Hatch and Slack. So it is also known as Hatch and Slack pathway. Okay, now moving ahead, photorespiration. I already told you what is photorespiration. Photorespiration is simply, it's a wasteful phenomena where rubisco enzyme, instead of binding with CO2, binds with O2, and instead of producing two molecules of phosphoglyceric acid, produce the first stable product, a 2-carbon compound phosphoglycolate, and one molecule of phosphoglyceric acid. Correct? Now, the last topic of the chapter is factors affecting photosynthesis. If I talk about the factors affecting photosynthesis, then those factors can be broadly divided into two categories, as internal or plant factors, and second are external factors like light, water, CO2, etc. Before we start with the factors affecting photosynthesis, students, there is one law which was proposed by scientist Blackman in the year 1905. Called as law of limiting factor. This law explains that if any chemical reaction is controlled by more than one factor, then the factor which is present in the least amount is going to decide the rate of the reaction. Like for example, if I talk in terms of photosynthesis, then this photosynthesis is governed by so many reactants like CO2, water, nutrients, etc., light, etc. Now, so many factors, when they are governing one chemical reaction, then the factor which is present in least amount, like for example, in case of photosynthesis, it is CO2. So, if CO2 get consumed up completely, no matter what is the amount of light available, no matter what is the amount of water available, photosynthesis will not operate. So, the rate of reaction is decided by the availability of the limiting factor, which is CO2 in case of photosynthesis. Correct? Chalo. Now, let's see some of the internal or plant factors which affect the rate of photosynthesis. Number one, very simple, is number of leaves on the plant body. You know, more the number of leaf, more pigments will be there, more light trapping will be done, and automatically rate of photosynthesis is also going to be high. Second, amount of chlorophyll in each leaf. Definitely, if amount of chlorophyll, if number of pigments in each leaf is high, then rate of photosynthesis is going to increase. Directly proportional. Third factor is age of the leaf. Younger the leaf, better its functions, better is the rate of photosynthesis. So you can say that age of leaf is inversely proportional to photosynthesis. How? Younger the leaf, more photosynthesis. Older the leaf, the leaf is about to senescence, it is losing its nutrient, the mobile nutrients are mobilizing from older to younger part. So definitely rate of photosynthesis is going to get affected adversely. So they are inversely proportional. Next is accumulation of product. What is the product of photosynthesis? Glucose. Now if already the leaf cells are having high amount of glucose, then why will they produce more glucose? If already food is there at your home, if already mother has prepared the food at home and it is in bulk, will you order more food from outside? No, because already food is so much at your home, you need not to order. So if already leaf cells are having high amount of sugar, if they have accumulated sugar, then that is going to reduce the rate of photosynthesis automatically. It is like a feedback inhibition. If product is already there, then it inhibits its own formation. Right? So these are the list of internal factors which you need to remember. Now, moving ahead with the external factors. The first external factor which I am going to cover is the major limiting factor, carbon dioxide. I already told you in the nature the amount of carbon dioxide present is between 0.03 to 0.04%, which is obviously very less if I talk in terms of light availability and water availability. That is why CO2 becomes one of the major limiting factor for photosynthesis. Right? So if concentration of CO2 rises for certain extent, maybe up to 0.05%, then rate of photosynthesis is also going to increase with the increase in CO2 concentration. Right? However, the response, now you have studied two groups of plant, C3 and C4. अब C3 में बेटा क्या है? They do not have internal CO2 concentration mechanism. They completely depend upon the external atmosphere for CO2. But if I talk about C4, you have seen right now, they show internal CO2 concentration mechanism. Correct? Though, so their dependence upon external environmental condition is slightly lesser as compared to C3 plant. So C4 plants get saturated sooner with respect to CO2 concentration as compared to C3. Chalo, let's check. So as I was telling you, C3 and C4 plants, they respond differently to CO2 concentration. Next, at low light intensity condition, obviously when another factor is less, when low light intensities are there and you keep on increasing the CO2 concentration, nothing is going to happen. Why? Because another factor is then going to become limiting. You make sure that with the increasing of CO2 concentration, light intensity is also increasing so that none of the factor become limiting and they keep on increasing the rate of photosynthesis. Right? However, at high light intensity, both C3 and C4 plant shows increase in the rate of photosynthesis, but it is important to know that C4 plants get saturated at 360 microns per liter. Their saturation rate is quicker. Why? Because they are already synthesizing CO2 inside them. However, if I talk about C4 plants, then they saturate at 450 microns, where it is written, yeah. Correct? So we can say that currently the environment is majorly limiting with respect to CO2 is more for C3 plants, because C3 plants are completely dependent upon the on the atmosphere for CO2. So definitely it is a major limiting factor for them. See, for plants, they are synthesizing their own CO2 inside. They are not that much dependent upon the atmosphere for CO2. So it may be limiting, but not as for C3. This much you have to do for CO2. Moving ahead, second is light. Light is also a very important external factor that governs the rate of photosynthesis. And sunlight can be studied on two parameters. The availability of light as an external factor affecting photosynthesis is dependent upon two natures of light. One is light quantity and second is light quality. You already know about action spectrum by T.W. Engelmann. You have seen that plant only prefers blue and red region of visible spectrum. You may give them green, orange, yellow. Photosynthesis rate will be lesser or no photosynthesis. But if you give them only blue and red region, plants are more than happy. That means quality matters but not quantity. You may give 100% of the light excluding blue and red. No photosynthesis. You only give them blue and red, maximum photosynthesis. So you can say that quality of light matters as plants only utilizes blue and red region of visible spectrum. Correct? Next is quantity. So quantity, again, I am going to explain this factor of light with the help of a very easy graph. So on the x-axis, we are going to mark the different wavelength of light or intensity, not wavelength of light, rather we should write intensity. Right? And here I am going to mark the rate of photosynthesis on y-axis. So when initially there is low light intensity and you increase the light intensity along with other factors, rate of photosynthesis is going to increase. Suppose initially there was very low light intensity, it is just the dawn time. It's early morning 4 o'clock. Now from 4 o'clock till 8 o'clock or till 9 o'clock, the light intensity is going to increase and surely the rate of photosynthesis will also increase. Right? After some time, after reaching to maximum rate of photosynthesis, even after increasing the amount of light, further rate of photosynthesis does not increase. That means some or the other factor has become limiting, maybe CO2. So there's a time or there's a peak of highlight intensity. Till that only photosynthesis rate is going to increase. Even after that, if you increase the rate of light, the rate of photosynthesis is going to become stagnant because other factors have become limiting. So rate of photosynthesis is constant. That means you have reached to the maximum level. But further now, if you increase the light intensity, then you're going to decrease the rate of photosynthesis due to solarization. Very high light intensity damages the chlorophyll pigment. And if chlorophyll pigments, they get damaged, then surely rate of photosynthesis is going to fall. So A to B is the line that shows increase in rate of photosynthesis. B to C is the line which shows constant photosynthesis, and C to D shows decline in the photosynthesis due to extreme high light intensity that causes solarization. So this is how the light intensity graph is. I hope light as an external parameter is also clear. Moving ahead, very simple, temperature and water. So first I'll talk about water. Usually water is not a limiting factor. Why? Because plant require very less, even less than 1% of the total amount of absorbed water for photosynthesis. No matter whatever is the whatever is the condition, maybe availability of water is poor in the soil, maybe plants are not able to transpire, but at least less than 1% of water will be always available for photosynthesis. If plant is absorbing 100% of water, then more than 99% is lost in the form of transpiration. Less than 1% is utilized in photosynthesis. So, no matter whatever is the amount of water in the soil, at least that 1% so will be available now, so that photosynthesis can properly run. So therefore, water availability never or usually never becomes a limiting factor, as the amount of required water is very less for photosynthesis. This much plant can always manage. Extreme condition you can remove, that there is drought for 2 years, for 3 years. Then that is a different case. Surely plant will die due to extreme lack of water. But little bit here and there, little bit change in the concentration of water in the soil, photosynthesis will not be affected. Transpiration can be affected, because of which absorption rate can be affected. But little bit absorption will always happen to just provide water for photosynthesis. So this much you have to remember for water. And if I talk about temperature, then there is optimum temperature for C3 plants and C4 plants, and under which the rate of photosynthesis is going to increase. But if you dip the temperature too low or you make the temperature too high, both the things are going to adversely affect the rate of photosynthesis. Very low temperature will freeze the system. Very high temperature will denature the protein, will affect negatively photosynthesis. So optimum temperature range is there for C3 and C4. For C4, that optimum range is slightly higher because they can tolerate high light intensities. And for C3, that optimum temperature range is slightly lower. Clear? So this much you have to do for temperature and water. I hope this concept is clear to each and every one of you. And with this, chapter photosynthesis is done. But before completing, let's try to do some questions out of it. First question says, primary acceptor of CO2 in C3 plant. So in C3 plant, the primary acceptor of CO2, I told you right now, is a 5-carbon compound, ribulose biphosphate. So please check what will be the answer. 2. RUBP. Yes? Correct. Moving ahead. Next question. Select the incorrect statement with respect to C4 plants. So you are given 4 statements and you have to tell which is correct and which is not correct. Number 1. Shows photorespiration. You know, C4 plants do not show photorespiration as they have their own internal CO2 concentration mechanism, which keeps the concentration of CO2 high in bundle sheath cell to avoid photorespiration. So first statement is incorrect. So here is the answer A. Right? Moving ahead. Next. Proton accumulates in which region of chloroplast for ATP synthesis? You remember chemiosmotic hypothesis by Peter Michel. In the lumen of the thylakoid, proton accumulates. Protons, they move from stroma in the lumen, because of which in the luminal space, proton accumulates. Proton concentration rises up, creating a gradient that is helpful for ATP synthesis. So the answer of the question becomes A. Hope you have understood this chapter well, and with this we come to the end of photosynthesis chapter. Thank you for joining me, guys. Keep in touch, keep studying, keep working hard, keep preparing. Thank you. All the best.